Welcome back to my YouTube channel. I'm nearly as popular as Joe Wicks now. Same beard, but less hair. Now, what I want you to do is, when you're doing these particular questions, I want you to take out the paper. I want you to read the paper. Then what I want you to do then I want you to watch the video. Pause the video wherever you want to. Go back, look at the question again, and forward the video again. Now, I had a student who asked me to look at a genetics question, especially the 2018 question. Now, it was question 13 that year, and the first part A, is name the biological active entities, each of which contains only one type of nucleic acid. That answer there is viruses. Viruses contain either DNA or RNA, never both. The second part is name two locations in eukaryotic cells where RNA but no DNA is found. The answer there is cytoplasm and the ribosome. Cytoplasm will contain transfer RNA, the ribosome will have messenger RNA and ribosomal RNA at it. Part B of that question, it basically is a genetic cross. And what you'll notice is from the actual question is there was a box, and the box had parental cross. It had round and wrinkled seeds, yellow and green seeds, purple and white flowers, tall and dwarf plants. Now that will become important here for part 2 of that particular question. Now what you were asked for is the law of segregation, which is the first of Mendel's laws. And that law is that traits are controlled by pairs of factors that separate a gamete formation. Now what that means is that we all have 2 traits for every single particular trait that's in our bodies. We get one from mommy and one from daddy. At gamete formation, one of those genes goes into one gamete, the other gene goes into another gamete. His second law is the law of independent assortment, and that states that each member of a pair of factors can combine with either member of another pair. Now what I mean by that is if you take these 2 chromosomes, I've got big T and small t, I've got big Y and small y. Big T can go with big Y. Likewise, big T can go with small y. And small t can go with big Y and small t can go with small y. So that's Mendel's second law. Now moving on then to the particular question. And the question on the paper was, a dwarf pea plant with green seeds was crossed with a plant heterozygous for both height and seed colour. So what I have here is, what you need to do first of all is write down with your hand. Big T is tall, small t is dwarf, big Y is yellow, and small y is green. So when I draw it out and I look at the parents of that, I've got a dwarf green plant and I've got a tall yellow plant, heterozygous. So here dwarf, which is small t, small y, which is green. Big T, small t, which is tall, big Y, small y, which is yellow. Now the gametes for that are small t and small y. Because over here, these are the chromosomes. Small t, which is dwarf, small y, which is green. Big T, small t, which is tall, big Y, small y, which is yellow. So here are my gametes for my dwarf green. Here is my tall and yellow. Now Mendel's second law is telling me this, that I can have big T and big Y. I can have big T and small y. But I can also have small t and big Y, and I can have small t and small y. Now, in order to get the genotypes of this particular cross, I need to draw my Punnett square. So my Punnett square is I'm going to put these gametes on top and this particular gamete here on the side. So here I've got small t and small y, big T, big Y, which I got here, big T, small y here, small t, big Y, small t, small y, which was here. So now I'm going to cross these two. So big T and small t, big Y and small y. Big T and small t, small y and small y. Small t and small t, big Y and small y. Small t, small t, small y and small y. What they look like or their phenotype are tall in yellow, tall in green, dwarf yellow, and dwarf in green. Now the next part of that question was, and the cross was, is if they are linked. What happens if they are linked? If those genes are linked, they are found on the same chromosome. Big Y and. So what happens here is, if they are linked, I have small t, small y here, but this time all that I have is big T and big Y, small t and small y. As you can see, I only have two gametes here. When they are linked, there are going to be two of them. When they were not linked, I had four. So therefore, I need to take these two away. So I've got my big T and big Y, small t and small y, crossed with my small t and small y. So I've got big T, small t, big Y, small y, which is tall in yellow. Here I have small t, small y, which is dwarf and green. Now they're looking exactly like the parents. So when they are linked, there is little variation. So when genes are linked, there's very, very little variation. Now tune in tomorrow for another question. Thank you.